Biomedical engineering. The fundamentals of biotechnology. Written by Phil Gilberts. Narrated by Kevin Brooker. 1. Introduction to biochemical engineering. Biochemical engineering integrates the principles of biology, chemistry, and engineering to develop processes for the large-scale production of valuable biochemicals. This interdisciplinary field plays a crucial role in industries such as pharmaceuticals, biotechnology, food and beverage, and environmental engineering. One of the fundamental aspects of biochemical engineering is fermentation, which involves the controlled growth of microorganisms like bacteria, yeast, or fungi to produce desired compounds. These microorganisms can be genetically engineered or selected for their ability to synthesize specific products, such as antibiotics, vitamins, or biofuels. Biocatalysis is another key area within biochemical engineering, focusing on the use of enzymes or whole cells to catalyze chemical reactions. By harnessing the specificity and efficiency of biological catalysts, biocatalysis offers environmentally friendly and cost-effective solutions for chemical synthesis and manufacturing processes. Bioseparation techniques are essential for isolating and purifying target biomolecules from complex mixtures produced during fermentation or biocatalytic reactions. These techniques include filtration, chromatography, centrifugation, and membrane processes, which enable the recovery of high-purity products for further downstream processing. Bioprocess design involves the optimization of various parameters such as reactor configuration, operating conditions, and media composition to maximize product yield, quality, and efficiency. Computational modeling and simulation techniques play a crucial role in predicting and optimizing bioprocess performance, allowing engineers to design robust and scalable production systems. Overall, biochemical engineering combines scientific principles with engineering methodologies to develop sustainable and economically viable processes for the production of biochemicals with diverse applications in medicine, agriculture, energy, and the environment. 2. Basics of biotechnology. Biotechnology is a broad field that applies biological principles and processes to develop products and technology that benefits society. Here are some basics. Definition: biotechnology involves the manipulation of living organisms or their components to create useful products and technologies. It encompasses various disciplines such as genetics, molecular biology, biochemistry, and microbiology. Applications: biotechnology has diverse applications in medicine, agriculture, food production, environmental remediation, and industrial processes. It has led to the development of new drugs, genetically modified crops, biofuels, biodegradable plastics, and diagnostic tools. Genetic engineering. Genetic engineering is a key aspect of biotechnology, involving the manipulation of an organism's genetic material to achieve desired traits or functions. Techniques like recombinant DNA technology and gene editing tools, such as CRISPR-Cas9, enable scientists to modify genes and create genetically modified organisms, or GMOs. Fermentation. Fermentation is a traditional biotechnological process used to produce various products such as beer, wine, cheese, and yogurt. It involves the controlled growth of microorganisms like yeast or bacteria to convert raw materials into useful compounds through biochemical reactions. Bioprocessing. Bioprocessing refers to the use of biological systems or components, such as enzymes or whole cells, to carry out chemical transformations or produce valuable products. It includes fermentation, biocatalysis, bioseparation, and downstream processing techniques to isolate and purify target compounds. Ethical and regulatory considerations. Biotechnology raises ethical and safety concerns, particularly regarding the use of genetically modified organisms and gene editing technologies. Regulatory frameworks exist to ensure the safety and ethical use of biotechnological products and practices. Future directions. Advances in biotechnology continue to drive innovation in fields such as personalized medicine, synthetic biology, gene therapy, and biomanufacturing. The integration of biotechnology with other disciplines, such as artificial intelligence and nanotechnology, holds promise for addressing complex societal challenges and improving human well-being. Understanding these basics provides a foundation for exploring the vast and rapidly evolving field of biotechnology and its impact on various aspects of society and the environment. 3. Cell biology and microbiology fundamentals. Cell biology and microbiology are foundational disciplines in the study of life sciences. Cell biology focuses on the structure, function, and behavior of cells, which are the basic units of life. Cells carry out essential processes such as metabolism, growth, reproduction, and response to stimuli. Key concepts in cell biology include cell organelles, such as the nucleus, mitochondria, and endoplasmic reticulum, cell membrane structure and function, cellular transport mechanisms, cell division, mitosis and meiosis, and cell signaling pathways. Understanding cell biology is crucial for various fields, including physiology, developmental biology, and pathology. Microbiology, on the other hand, is the study of microorganisms, which include bacteria, viruses, fungi, protozoa, and algae. Microorganisms play diverse roles in nature, ranging from beneficial symbiotic relationships to causing infectious diseases. Microbiology encompasses various subdisciplines, such as bacteriology, virology, mycology, and parasitology. Key topics in microbiology include microbial morphology, genetics, metabolism, ecology and pathogenesis. Microbiologists study microbial diversity, interactions between microorganisms and their environments, microbial evolution, and the development of antimicrobial strategies. Both cell biology and microbiology are interdisciplinary fields that contribute to our understanding of fundamental biological processes and their applications in medicine, biotechnology, agriculture, and environmental science. They provide insights into the complexities of life at the cellular and microbial levels and offer opportunities for innovation and discovery in scientific research and technological development. 4. Enzyme kinetics and catalysis. Enzyme kinetics is a study of the rates at which enzymes catalyze chemical reactions. Enzymes are biological catalysts that accelerate chemical reactions by lowering the activation energy required for the reaction to occur. Understanding enzyme kinetics is essential for elucidating the mechanisms by which enzymes function and for optimizing enzyme-catalyzed reactions in various applications. The Michaelis-Menten equation is a fundamental concept in enzyme kinetics, describing the relationship between the rate of an enzyme-catalyzed reaction, or V, and the concentration of substrate, or S. According to the Michaelis-Menten model, the initial reaction rate, VO, is directly proportional to the concentration of enzyme substrate complex, or ES, which is formed when the enzyme binds to the substrate. Other models, such as the Lineweaver-Burke plot and the Edey-Hofstee plot, can also be used to analyze enzyme kinetics data and determine kinetic parameters. Enzyme catalysis involves several steps, including substrate binding to the enzyme's active site, formation of the enzyme substrate complex, catalytic conversion of the substrate into product, and release of the product. The specificity and efficiency of enzyme catalysis depend on factors such as enzyme-substrate interactions, conformational changes in the enzyme, and the presence of cofactors or coenzymes. Summarized, enzyme kinetics and catalysis are crucial areas of study in biochemistry and biotechnology, with applications in drug discovery, enzyme engineering, metabolic engineering, and industrial biocatalysis. Understanding the mechanisms of enzyme action allows scientists and engineers to design and optimize enzymatic processes for various practical purposes. 5. Metabolic pathways and regulation. Metabolic pathways are a series of interconnected biochemical reactions that convert substrates through a sequence of intermediates into final products. These pathways are essential for maintaining the cell's energy balance, building cellular structures, and enabling cell growth and division. Metabolic pathways can be categorized into catabolic pathways, which break down molecules to release energy, and anabolic pathways, which use energy to synthesize complex molecules from simpler ones. One of the central catabolic pathways is glycolysis, where glucose is broken down into pyruvate, generating ATP and NADH. This is followed by the citric acid cycle, or Krebs cycle, which further oxidizes the products of glycolysis to produce more ATP, NADH, and FADH2. These electron carriers then donate electrons to the electron transport chain in the process of oxidative phosphorylation, producing a large amount of ATP. These pathways highlight the interconnected nature of cellular respiration, which efficiently converts energy stored in organic molecules into usable cellular energy. An example of an anabolic pathway is the Kelvin cycle in photosynthesis, where carbon dioxide is fixed into glucose using the energy from ATP and NADPH produced in the light reactions of photosynthesis. Similarly, the synthesis of amino acids, nucleotides, and fatty acids are vital anabolic processes, each involving multiple enzymatic steps and intricate regulation. Regulation of metabolic pathways ensures that cells respond to changes in their internal and external environments and maintain homeostasis. This regulation occurs at multiple levels, including 1. Enzyme regulation. Enzyme activity can be regulated through allosteric regulation, where an effector molecule binds to an enzyme at a site other than the active site, causing a conformational change that affects its activity. Enzyme activity can also be controlled by covalent modifications, such as phosphorylation, which can activate or deactivate enzymes. 2. Gene expression. The amount of enzymes present in the cell can be regulated at the transcriptional level. Genes encoding enzymes involved in metabolic pathways can be up-regulated or down-regulated in response to environmental signals or cellular needs. 3. Substrate availability. The concentration of substrates can influence the rate of metabolic reactions. An increase in substrate concentration typically enhances the reaction rate until the enzymes become saturated. 4. Feedback inhibition. In many metabolic pathways, the end product of the pathway can inhibit an earlier step in the pathway, providing a feedback mechanism to prevent the overproduction of the final product. 5. Compartmentalization. Eukaryotic cells compartmentalize certain metabolic pathways within specific organelles. For example, the citric acid cycle occurs in the mitochondria, while fatty acid synthesis occurs in the cytoplasm, allowing distinct regulation of these pathways. An example of pathway regulation is the control of glycolysis and gluconeogenesis. Glycolysis is regulated by the availability of glucose and the energy needs of the cell, with key regulatory enzymes like hexokinase, phosphofructokinase, and pyruvate kinase being tightly controlled. In contrast, gluconeogenesis, the synthesis of glucose from non-carbohydrate sources, is activated when there is a need to produce glucose, such as during fasting. Overall, metabolic pathways and their regulation are crucial for cellular function and adaptation, allowing organisms to efficiently manage resources, respond to environmental changes, and maintain homeostasis. Understanding these pathways and their control mechanisms is fundamental in fields such as medicine, biotechnology, and metabolic engineering. 6. Microbial cultivation techniques. Microbial cultivation techniques are essential for studying microorganisms and utilizing them in various applications, including research, industrial production, and clinical diagnostics. These techniques involve growing and maintaining microorganisms under controlled conditions to ensure optimal growth and productivity. Here are some key microbial cultivation techniques. 1. Culture media. Culture media provide the necessary nutrients for microbial growth. They can be classified into several types. Liquid media, or broths, used for growing large quantities of microorganisms in a homogeneous environment. Solid media, agar platies, used for isolating and counting microorganisms, as well as for observing colony morphology. Agar, a gelatinous substance, is added to the nutrient medium to solidify it. Selective media contains specific substances that favor the growth of certain microorganisms while inhibiting others. For example, McConkey agar is selective for gram-negative bacteria. Differential media contain indicators that allow differentiation between different types of microorganisms based on their metabolic activities. Blood agar can differentiate bacteria based on their hemolytic properties. 2. Sterilization. Ensuring that culture media and equipment are free from contaminants is crucial. Common sterilization methods include autoclaving. Uses high-pressure steam to sterilize media and equipment. Filtration. Sterilizes heat-sensitive liquids by passing them through a filter with pores small enough to remove microorganisms. Chemical sterilization. Uses chemicals like ethanol or bleach to disinfect surfaces and equipment. 3. Inoculation techniques. Inoculation introduces microorganisms into culture media. Techniques include streak plate method, used to isolate pure colonies from a mixed sample by streaking the inoculum over the surface of an agar plate. Pour plate method involves mixing the inoculum with molten agar and pouring it into a Petri dish to allow colonies to grow within the medium. Spread plate method. Spreading a liquid sample evenly over the surface of an agar plate using a sterile spreader. Loop dilution. Serially diluting a microbial sample in liquid media to achieve a range of concentrations for quantitative studies. 4. Incubation conditions. Proper incubation conditions are critical for microbial growth, including temperature. Different microorganisms have optimal growth temperatures such as mesophiles at 20 to 45 degrees Celsius, thermophiles above 45 degrees Celsius, and psychrophiles below 20 degrees Celsius. Aeration. Some microorganisms require oxygen, or aerobies, while others grow in its absence, anaerobies. Facultative anaerobies can grow with or without oxygen. pH. The pH of the culture medium must be appropriate for the microorganism being cultivated, with most bacteria preferring neutral pH, while fungi often thrive in slightly acidic conditions. 5. Monitoring growth. Monitoring microbial growth helps assess culture health and productivity. Turbidity measurement. Uses a spectrophotometer to measure the cloudiness of a liquid culture, which correlates with cell density. Viable count. Determines the number of living cells by counting colony-forming units, or CFUs, on agar plates. Direct microscopic counts. Counts cells directly under a microscope using a counting chamber. 6. Continuous culture systems. Continuous culture techniques, such as chemostats and bioreactors, maintain microbial growth in a steady state by continuously supplying fresh nutrients and removing waste. This allows for the study of microbial physiology under controlled conditions and the production of metabolites on an industrial scale. Microbial cultivation techniques are fundamental for studying microbial physiology, genetics, and interactions, as well as for developing biotechnological applications such as the production of antibiotics, enzymes, and biofuels. These techniques enable scientists to manipulate and utilize microorganisms effectively for a wide range of scientific and industrial purposes. 7. Downstream processing and separation techniques. Downstream processing is a crucial aspect of biotechnology and biochemical engineering, involving the recovery and purification of biosynthetic products, particularly pharmaceuticals, from natural resources such as microbial fermentation, cell cultures, or plant tissues. The aim is to obtain a product in its purest form, suitable for its intended use. The process consists of several stages, each involving different separation techniques. Here are the key stages and techniques used in downstream processing. 1. Cell disruption. Mechanical methods. Homogenization. Using high-pressure homogenizers to break cells' walls by forcing the cell suspension through a narrow orifice. Bead milling. Grinding cells with small beads in a milling machine to rupture them through shear and collision forces. Ultrasonication. Applying ultrasonic waves to create cavitation and shear forces that disrupt cell membranes. Non-mechanical methods. Chemical lysis. Using detergents, solvents, or enzymes to dissolve cell walls and membranes. Osmotic shock. Exposing cells to a sudden change in osmotic pressure, causing them to burst. 2. Solid-liquid separation. Centrifugation. using centrifugal force to separate cellular components based on their size and density. It can be differential centrifugation, separates components by successive rounds of centrifugation at increasing speeds, or density gradient centrifugation, which uses a gradient of a dense substance, like sucrose, to separate components based on their buoyant density. Filtration, employing physical barriers to separate solids from liquids. It includes depth filtration, uses a thick filter medium to capture particles throughout its depth. Membrane filtration, uses a thin membrane with specific pore sizes for more precise separation. 3. Product isolation. Precipitation, adding salts or salting out, solvents or solvent precipitation, or changing pH to precipitate proteins or other biomolecules from the solution. Extraction, using solvents to selectively dissolve and extract the desired product. Types include liquid-liquid extraction, separating components based on their solubility in two immiscible liquids. Solid-liquid extraction, extracting components from a solid matrix using a liquid solvent. 4. Product purification. Chromatography, ion exchange chromatography, separates molecules based on their charge using a charged resin. Affinity chromatography, uses a specific binding interaction, e.g. antibody-antigen, to isolate the target molecule. Size exclusion chromatography, or gel filtration, separates molecules based on size using a porous gel matrix. Hydrophobic interaction chromatography, separates molecules based on their hydrophobicity. Electrophoresis, separates molecules based on their charge and size by applying an electric field. It includes techniques like SDS-PAGE for proteins and agarose gel electrophoresis for nucleic acids. 5. Final polishing, ultrafiltration and diafiltration, using semi-permeable membranes to concentrate and further purify proteins or other macromolecules by removing small impurities. Crystallization, inducing the formation of crystals from a purified solution to obtain the product in a solid and stable form. 6. Formulation and packaging. After purification, the product must be formulated with stabilizers, preservatives, and other excipients to ensure its stability, efficacy, and shelf life. The final step involves packaging the product in a suitable format for storage, distribution, and use. Downstream processing is a multi-step, complex, and crucial phase in the production of biotechnological products. The selection of appropriate techniques depends on the nature of the product, its intended use, and the required purity levels. Mastery of these techniques ensures the efficient, cost-effective, and safe production of high-quality bioproducts, meeting both regulatory standards and market demands. 8. Bioreactor design and operation. Bioreactors are vessels in which biological reactions occur, typically involving microorganisms, animal cells, or plant cells, to produce a desired product such as pharmaceuticals, enzymes, biofuels, or chemicals. The design and operation of bioreactors are critical for optimizing production, ensuring process stability, and maintaining product quality. Here are the key aspects of bioreactor design and operation. Bioreactor design. Types of bioreactors. Stirred-tank bioreactors, or STBR. The most common type, featuring a cylindrical vessel with an impeller for mixing and aeration. They are versatile and suitable for both microbial and cell culture processes. Airlift bioreactors. Utilize a draft tube to circulate fluids and maintain aeration without mechanical stirring, ideal for shear-sensitive cultures. Packed bed bioreactors. Contain immobilized cells on a solid matrix, often used in continuous processes and for products requiring high cell densities. Fluidized bed bioreactors. Similar to packed beds, but with fluid flow that suspends the solid particles, providing better mass transfer. Wave bioreactors. Using a rocking motion to mix and aerate the culture, particularly useful for mammalian cell cultures. Key components. Vessel. Made of materials like stainless steel or glass for durability and sterility. Impeller. For mixing and maintaining homogeneous conditions. Different types include Russian turbines, marine impellers, and pitched blade turbines. Sparger. Introduces air or oxygen into the culture medium, essential for aerobic processes. Sensors and probes. Monitor critical parameters such as pH, dissolved oxygen or DO, temperature, and biomass concentration. Control systems. Automated systems to maintain optimal conditions, including feedback mechanisms to adjust pH, DO, temperature, and nutrient feed. Skill and configuration. Lab scale. Small volumes up to a few liters for research and development. Pilot scale. Intermediate volumes, tens to hundreds of liters, for process optimization and scale-up studies. Production scale. Large volumes, thousands to tens of thousands of liters, for commercial production. Bioreactor operation. Operating modes. Batch. All ingredients are added at the start, and the culture is allowed to grow until harvest. Simple to operate, but can be less efficient. Fed batch. Nutrients are added periodically without removing culture fluid, allowing higher cell densities and product yields. Continuous. Fresh medium is continuously added and culture fluid is continuously removed, maintaining a steady state condition. Offers high productivity and consistent product quality, but requires sophisticated control systems. Key operational parameters. Temperature must be controlled to optimize enzyme activity and cell growth, typically maintained within a narrow range specific to the organism. pH maintained using buffers or automatic pH control systems to ensure optimal enzyme activity and cell health. Dissolved oxygen, or DO. Critical for aerobic processes maintained by adjusting aeration rates using oxygen-enriched air or controlling agitation speed. Nutrient supply. Proper nutrient levels are critical for growth and productivity, often controlled through feed strategies in fed batch and continuous systems. Agitation and mixing. Ensure homogeneous conditions, prevent cell settling, and enhance mass transfer. Agitation speed must be optimized to balance mixing efficiency and shear stress. Sterilization. In situ sterilization. Autoclaving the entire bioreactor and associated piping. CIP, or clean in place, and SIP, or sterilize in place. Automated cleaning and sterilization processes to maintain sterility between batches without disassembling the bioreactor. Scale-up considerations. Geometric similarity. Maintaining similar vessel dimensions and aspect ratios when scaling from lab to production scale. Dynamic similarity. Ensuring similar mixing and mass transfer conditions by adjusting impeller speeds and aeration rates based on power input per unit volume and oxygen transfer rates. Designing and operating bioreactors involves a deep understanding of both biological and engineering principles. Effective bioreactor design ensures optimal conditions for cell growth and product formation, while efficient operation strategies maximize yield and productivity. As bioprocesses move from lab scale to industrial scale production, considerations of scalability, control, and sterility become increasingly critical, necessitating sophisticated design and automation solutions. 9. Genetic engineering and recombinant DNA technology. Genetic engineering and recombinant DNA technology are transformative fields in modern biology, enabling precise modifications to the genetic material of organisms for various applications, from medical therapies to agricultural improvements and industrial biotechnology. Here's a detailed look at these concepts. Genetic engineering. Definition. Genetic engineering involves the direct manipulation of an organism's genome using biotechnology to alter its characteristics. This can include adding, removing, or modifying genes to achieve desired traits. Applications. Medicine. Development of gene therapies, production of insulin, human growth hormones, and vaccines. Agriculture. Creation of genetically modified crops with improved yield, pest resistance, and nutritional content. Industrial biotechnology. Production of enzymes for detergents, biofuels, and biodegradable plastics. Research. Creation of model organisms to study gene function and disease. Recombinant DNA technology. Definition. Recombinant DNA technology involves combining DNA from different sources to create sequences that do not naturally occur. This process typically involves isolating a gene of interest and inserting it into a vector for propagation and expression in a host organism. Key steps. Gene isolation. Restriction enzymes. Enzymes that cut DNA at specific sequences, generating fragments with sticky or blunt ends. Polymerase chain reaction, or PCR. Amplifies specific DNA sequences, making it easier to work with large amounts of a target gene. Vector preparation. Plasmids. Circular DNA molecules used as vectors to carry foreign genes into host cells. They often contain selectable markers, e.g., antibiotic resistance genes, to identify successful transformants. Viral vectors. Viruses engineered to deliver genetic material into cells, often used in gene therapy. Other vectors. Cosmids, BACs, or bacterial artificial chromosomes, and YACs, or yeast artificial chromosomes, are used for larger DNA fragments. Ligation. DNA ligase. Enzyme that joins DNA fragments together by forming covalent bonds between the sugar phosphate backbones of DNA strands. Transformation. Bacterial transformation. Introduction of recombinant DNA into bacteria using methods like heat shock or electroporation. Transfection. Introduction of recombinant DNA into eukaryotic cells using chemical, lipid-based, or physical methods. Selection and screening. Antibiotic selection. Use of antibiotics to select for cells that have taken up the plasmid with the antibiotic resistance gene. Reporter genes. Genes that encode easily detectable proteins, e.g., GFP, green fluorescent protein, used to identify successful recombinant events. Expression. Expression vectors. Contain promoters and other regulatory elements to ensure the foreign gene is transcribed and translated within the host organism. Host systems. E. coli, yeast, insect cells, mammalian cells, and plants, each offering different advantages depending on the protein's complexity and post-translational modifications required. Techniques and tools. CRISPR-Cas9. A revolutionary genome editing tool that allows for precise, targeted changes to the DNA of living organisms. It uses a guide RNA to direct the Cas9 enzyme to a specific DNA sequence where it makes a cut, allowing for the addition or removal of genetic material. Gene cloning. Creating copies of a specific gene by inserting it into a plasmid vector and introducing it into a bacterial host. The bacteria replicate, producing many copies of the gene. Gel electrophoresis. Technique to separate DNA fragments by size, allowing for the analysis and purification of DNA. Sequencing. Determining the exact sequence of nucleotides in a DNA molecule. Techniques like Sanger sequencing and next-generation sequencing, or NGS, are used for various applications, from identifying genetic disorders to studying biodiversity. Ethical and regulatory considerations. Genetic engineering and recombinant DNA technology raise ethical questions about safety, environmental impact, and the morality of altering life forms. Regulatory bodies like the FDA, or Food and Drug Administration, and EPA, or Environmental Protection Agency, in the United States, as well as international organizations, establish guidelines and regulations to ensure the safe and ethical use of these technologies. Genetic engineering and recombinant DNA technology are pivotal in advancing our understanding of biology and enhancing our ability to manipulate genetic material for a wide range of applications. From improving healthcare and agriculture to facilitating scientific research, these technologies continue to shape the future of science and society. 10. Fermentation processes. Fermentation processes are biological reactions where microorganisms, such as bacteria, yeast, or fungi, convert organic compounds, usually carbohydrates, into alcohol or acids under anaerobic conditions. These processes are central to various industries, including food and beverage production, pharmaceuticals, and biofuels. Here's a detailed overview of fermentation processes. Types of fermentation. Alcoholic fermentation. Microorganisms, primarily yeast. Saccharomyces cerevisiae. Substrate, sugars, glucose, fructose, sucrose. Products, ethanol and carbon dioxide. Applications, brewing beer, winemaking, and production of bioethanol as a renewable fuel. Lactic acid fermentation. Microorganisms, lactic acid bacteria, Lactobacillus streptococcus. Substrate, sugars. Products, lactic acid. Applications, production of yogurt, sauerkraut, kimchi, and certain cheeses, as well as lactic acid for industrial use. Acetic acid fermentation. Microorganisms, acetic acid bacteria, Acetobacter. Substrate, ethanol. Products, acetic acid. Applications, production of vinegar. Butyric acid fermentation. Microorganisms, Clostridium species. Substrate, sugars and starches. Products, butyric acid, carbon dioxide, and hydrogen. Applications, production of solvents and biofuels. Mixed acid fermentation. Microorganisms, enteric bacteria, Escherichia coli. Substrate, sugars. Products, a mixture of acids, lactic acetic, formic, ethanol, carbon dioxide, and hydrogen. Applications: understanding metabolic pathways in bacteria and industrial solvent production. Fermentation processes: batch fermentation. Operation: all substrates and microorganisms are added at the beginning, and the process runs until the nutrients are exhausted. Advantages: simple to set up and operate, suitable for high-value products. Disadvantages: limited by substrate concentration, potential for inconsistent product quality. Fed batch fermentation. Operation: substrates are added in increments during the fermentation process, allowing for extended fermentation and higher product yields. Advantages: better control over growth conditions, higher cell densities, and product concentrations. Disadvantages: more complex operation and control systems. Continuous fermentation. Operation: fresh substrate is continuously added and culture fluid is continuously removed to maintain a steady state. Advantages: consistent product quality, high productivity, and efficient use of fermenter volume. Disadvantages: complex control systems, risk of contamination. Fermentation parameters. Temperature. Optimal temperature varies by microorganism. Must be controlled to maximize microbial activity and product yield. pH. The pH of the medium affects enzyme activity and microorganism health. Buffers or automatic pH control systems are used to maintain optimal pH levels. Aeration and oxygen supply. Critical for aerobic fermentation processes. Oxygen is sparged into the fermenter, and agitation ensures proper mixing and oxygen transfer. Agitation. Mechanical stirring or bubbling air to keep the culture homogeneous and enhance mass transfer of nutrients and gases. Nutrient supply. Sufficient nutrients must be provided to support microbial growth and product formation. In fed batch and continuous systems, nutrient addition is carefully controlled. Sterility. Maintaining a sterile environment is crucial to prevent contamination. Sterilization of the fermenter, media, and air supply is typically achieved through autoclaving or filtration. Fermentation equipment. Fermenters or bioreactors. Vessels designed for controlled fermentation, available in various scales from laboratory to industrial. Stirred tank fermenters or STF. Common for microbial fermentations, equipped with agitators and spargers. Airlift fermenters. Used for shear-sensitive cultures, employing air circulation for mixing. Packed bed and fluidized bed reactors. Used for immobilized cell systems, where cells are attached to a solid support. Control systems. Automated systems to monitor and adjust parameters such as temperature, pH, dissolved oxygen, and nutrient feed, ensuring optimal fermentation conditions. Applications of fermentation. Food and beverage industry. Production of bread, beer, wine, yogurt, cheese, and fermented vegetables. Pharmaceutical industry. Production of antibiotics, penicillin, streptomycin, vaccines, and therapeutic proteins, insulin, or growth hormones. Biofuel production. Conversion of biomass into bioethanol and biobutanol. Industrial biotechnology. Production of organic acids, citric acid, lactic acids, solvents, acetone, butanol, and enzymes, amylases, proteases. Fermentation processes are versatile and widely used in various industries to produce valuable products. Understanding the different types of fermentation, operational modes, and key parameters is essential for optimizing production and achieving consistent, high-quality outputs. Advances in fermentation technology continue to drive innovation in biotechnology, offering sustainable solutions for food, medicine, and energy. 11. Bioprocess control and optimization. Bioprocess control and optimization are essential elements in industrial biotechnology, focusing on maximizing efficiency, yield, and quality of biological products. This involves regulating various parameters during bioprocesses such as microbial fermentation, cell culture, and enzymatic reactions. Bioprocess control involves monitoring and regulating the physical, chemical, and biological parameters of a bioprocess to maintain optimal conditions for the production of the desired product. Key parameters include temperature, pH, dissolved oxygen or DO, agitation and mixing, nutrient supply, and foam control. Temperature control is vital as it affects enzyme activity and microbial growth rates. Automated temperature control systems use thermocouples or resistance temperature detectors, or RTDs, linked to heating and cooling systems. pH is another critical parameter influencing enzyme activity, cell metabolism, and product stability, maintained using automated pH control systems with pH probes and acid or base solutions. Dissolved oxygen is crucial for aerobic microorganisms and cell cultures, ensured by DO sensors coupled with air or oxygen spargers and agitation control systems. Agitation and mixing are essential for a homogeneous distribution of nutrients, gases, and cells, achieved through mechanical stirrers or airlift systems with adjustable speeds to optimize mixing without causing shear stress. Proper nutrient supply is critical for cell growth and product formation, controlled through fed batch and continuous feeding strategies based on monitoring self-growth and nutrient concentration. Foam control is necessary as excessive foam can interfere with oxygen transfer and cause contamination, managed by anti-foam agents or mechanical foam breakers controlled by foam sensors. Monitoring techniques are vital for effective bioprocess control, encompassing in situ, online, and offline methods. In situ monitoring includes measuring optical density, or OD, to assess cell concentration by absorbance, spectroscopy to monitor metabolites and product concentrations, and flow cytometry to analyze cell size, viability, and expression levels. Online monitoring employs biosensors to detect specific metabolites, nutrients, or environmental conditions, and mass spectrometry for real-time analysis of fermentation products and byproducts. Offline monitoring involves periodic sampling and analysis using lab-based assays like HPLC or high-performance liquid chromatography, or GC, gas chromatography. Bioprocess optimization aims to improve the efficiency and productivity of the bioprocess through systematic adjustments and enhancements. This includes the use of design of experiments, or DOE, to systematically investigate the effects of multiple factors on the bioprocess and identify optimal conditions, employing methods such as factorial designs, response surface methodology, or RSM, and Taguchi methods. Mathematical modeling and simulation develop mathematical representations of bioprocesses to predict outcomes and optimize parameters, using kinetic models, metabolic flux analysis, and computational fluid dynamics, or CFD. Adaptive control systems automatically adjust process parameters in real time based on feedback from monitoring systems, employing model predictive control, or MPC, fuzzy logic control, and neural networks. Scale-up and scale-down studies ensure that bioprocesses are scalable from laboratory to industrial production while maintaining performance and product quality, conducting trials at different scales and using data to inform process adjustments. Optimization of media and feed strategies enhances cell growth and product yield by optimizing nutrient composition and feeding regimens, using DOE and empirical optimization. Advanced control techniques include process analytical technology, or PAT, and quality by design, or QDB. And so, while PAT uses real-time data and advanced analytics to control and optimize bioprocesses, incorporating tools like spectroscopy, chromatography, and advanced sensors integrated with data analysis software, QDB is a systematic approach to bioprocess development, emphasizing understanding and controlling variability to ensure product quality, and implemented by identifying critical quality attributes, or CQAs, and critical process parameters, or CPPs. It also establishes control strategies. 12. Applications of biochemical engineering in industry. Biochemical engineering applies principles of biology, chemistry, and engineering to develop and optimize processes involving biological organisms or molecules. This interdisciplinary field has numerous applications across various industries, driving innovations and improving efficiency and sustainability. Here are some key applications of biochemical engineering in industry. Pharmaceuticals and biotechnology. Biochemical engineering plays a pivotal role in the development and manufacturing of pharmaceuticals. It is instrumental in producing antibiotics, vaccines, hormones, and therapeutic proteins. For instance, recombinant DNA technology allows for the production of insulin and monoclonal antibodies. Bioprocess engineers design and optimize fermentation processes and cell culture systems to ensure high yields, product purity, and cost effectiveness. Advanced techniques such as continuous bioprocessing and single-use bioreactors are increasingly adopted to enhance production flexibility and scalability. Food and beverage industry. The food and beverage industry extensively uses biochemical engineering to improve food quality, safety, and nutritional value. Fermentation processes are employed to produce a variety of foods, including yogurt, cheese, bread, beer, and wine. Bioprocess engineers optimize fermentation conditions to enhance flavor, texture, and shelf life. Enzyme technology is also crucial, enabling the production of high fructose corn syrup, lactose-free dairy products, and improved bread-making processes. Additionally, metabolic engineering of microorganisms is used to produce food additives and nutraceuticals. Biofuels and bioenergy. In the quest for sustainable energy sources, biochemical engineering is vital for the production of biofuels such as bioethanol, biodiesel, and biogas. Engineers develop and optimize microbial fermentation processes to convert biomass, including agricultural residues and algae, into fuels. Advances in metabolic engineering and synthetic biology allow for the development of microorganisms with enhanced capabilities to produce biofuels more efficiently. Biochemical engineers also work on integrating biofuel production with other industrial processes to improve overall energy efficiency and reduce waste. Environmental engineering. Biochemical engineering contributes to environmental protection and sustainability through bioremediation, waste treatment, and pollution control. Bioremediation uses microorganisms to degrade environmental contaminants, such as oil spills, heavy metals, and organic pollutants. Waste treatment processes, including anaerobic digestion and composting, convert organic waste into valuable products like biogas and compost. Biochemical engineers design and optimize these processes to maximize efficiency and minimize environmental impact. Additionally, bio-based technologies are developed to produce biodegradable plastics and reduce reliance on petrochemicals. Agricultural biotechnology. The agricultural sector benefits significantly from biochemical engineering through the development of genetically modified crops and biopesticides. Genetically engineered crops can have improved traits such as pest resistance, herbicide tolerance, and enhanced nutritional content. Bioprocess engineers work on producing biofertilizers and biopesticides using microbial fermentation, which can reduce the use of chemical fertilizers and pesticides promoting sustainable agriculture. Furthermore, advances in plant tissue culture and bioreactor design facilitate the large-scale production of high-value crops and plant-derived compounds. Industrial biotechnology. In industrial biotechnology, biochemical engineering is essential for the production of chemicals, materials, and enzymes through bioprocesses. Microbial fermentation and enzyme catalysis are used to produce a wide range of chemicals, including organic acids, e.g., citric acid, lactic acid, solvents, e.g., ethanol, acetone, and biopolymers, e.g., polylactic acid. Bioprocess engineers optimize these production processes to increase yields, reduce costs, and improve sustainability. Enzyme technology also plays a crucial role in industrial processes, such as textile treatment, paper bleaching, and detergent formulation, enhancing efficiency and reducing environmental impact. Medical devices and diagnostics. Biochemical engineering contributes to the development of advanced medical devices and diagnostic tools. This includes the design and production of biosensors for detecting diseases, monitoring health conditions, and ensuring food safety. Biochemical engineers work on developing biocompatible materials and coatings for implants and prosthetics. Additionally, lab-on-a-chip technologies and microfluidic devices are engineered to enable rapid point-of-care diagnostics, improving healthcare accessibility and outcomes. Cosmetics and personal care. The cosmetics and personal care industry utilizes biochemical engineering to develop and manufacture products such as skincare creams, shampoos, and cosmetics. Enzymes and biotechnologically derived ingredients such as hyaluronic acid, collagen, and peptides are used to enhance product efficacy and safety. Biochemical engineers optimize the production processes for these ingredients, ensuring consistency and quality. Additionally, sustainable practices such as using bio-based and biodegradable materials are increasingly adopted to reduce environmental impact. Biochemical engineering is a cornerstone of numerous industries, driving innovation and sustainability. Its applications range from pharmaceuticals and food production to biofuels and environmental protection. By integrating biological principles with engineering practices, biochemical engineers develop efficient, cost-effective, and sustainable processes that address global challenges and improve the quality of life. As technology advances, the role of biochemical engineering will continue to expand, opening new possibilities and contributing to the development of a more sustainable future. 13. Future trends in biotechnology. The future of biotechnology holds exciting advancements and transformative potential across various fields. Emerging trends are driven by technological innovations, interdisciplinary research, and a growing focus on sustainability and health. Here are some key future trends in biotechnology. Synthetic biology. Synthetic biology is poised to revolutionize biotechnology by enabling the design and construction of new biological parts, devices, and systems. This field combines principles from biology, engineering, and computer science to create synthetic organisms with tailored functions. Future trends in synthetic biology include gene editing and CRISPR. The use of CRISPR-Cas9 and other gene editing tools to precisely modify genomes will expand, allowing for the creation of organisms with desirable traits, such as disease resistance or enhanced metabolic capabilities. Biosensors. Development of biosensors for environmental monitoring, healthcare diagnostics, and industrial applications will grow, providing real-time detection of specific molecules or pathogens. Biomanufacturing. Engineered microbes will be used to produce complex chemicals, pharmaceuticals, and materials, offering sustainable alternatives to traditional manufacturing processes. Personalized medicine. Personalized medicine aims to tailor medical treatments to individual patients based on their genetic, environmental, and lifestyle factors. Advances in genomics, bioinformatics, and biotechnology are driving this trend. Genomic sequencing. Rapid and cost-effective genome sequencing will become more accessible, enabling detailed genetic profiling of patients. Pharmacogenomics. Understanding how genetic variations affect drug response will lead to the development of personalized therapies, reducing adverse effects and improving efficacy. Biomarker discovery. Identification of new biomarkers will facilitate early diagnosis and targeted treatment of diseases, particularly in oncology. Regenerative medicine. Regenerative medicine focuses on repairing or replacing damaged tissues and organs through the use of stem cells, tissue engineering, and biomaterials. Key trends in this field include stem cell therapy. Advances in stem cell technology will lead to new treatments for conditions such as neurodegenerative diseases, heart disease, and spinal cord injuries. 3D bioprinting. The use of 3D printing to create tissue and organ constructs will progress, potentially leading to the fabrication of functional organs for transplantation. Organoids. Miniature, lab-grown organ models will be used for drug testing, disease modeling, and personalized medicine, providing insights into complex biological processes. Agricultural biotechnology. Agricultural biotechnology is essential for enhancing food security, sustainability, and crop resilience. Future trends in this area include genome editing in crops. Techniques like CRISPR will be used to develop crops with improved traits such as drought tolerance, pest resistance, and higher nutritional value. Microbiome engineering. Understanding and manipulating the plant microbiome will enhance crop health and productivity, reducing the need for chemical fertilizers and pesticides. Vertical farming and controlled environment agriculture, or CEA. Innovations in indoor farming technologies will optimize resource use and enable year-round production of high-quality crops in urban settings. Industrial biotechnology. Industrial biotechnology aims to develop sustainable processes for the production of chemicals, materials, and biofuels. Key future trends include bio-based materials, development of bio-based plastics, textiles, and composites will reduce reliance on fossil fuels and decrease environmental impact. Biorefineries. Integrated biorefineries will convert biomass into a range of products, including biofuels, chemicals, and bioproducts, enhancing the efficiency and economics of bio-based production. Enzyme engineering. Advances in enzyme engineering will create more robust and efficient biocatalysts for industrial processes, improving sustainability and reducing costs. Environmental biotechnology. Environmental biotechnology focuses on using biological processes for environmental restoration and sustainability. Future trends include bioremediation. Enhanced techniques for bioremediation will address pollution and contamination in soil, water, and air, using engineered microorganisms to degrade harmful substances. Waste-to-resource technologies. Innovations will convert waste materials into valuable products such as bioenergy, bioplastics, and fertilizers, promoting a circular economy. Carbon sequestration. Biological methods for capturing and storing carbon dioxide will be developed to mitigate climate change, including engineered plants and microbes designed to sequester carbon more efficiently. Computational biology and bioinformatics. Computational biology and bioinformatics are integral to advancing biotechnology through data analysis, modeling, and simulation. Future trends include artificial intelligence, or AI, and machine learning. AI and machine learning will revolutionize data analysis, enabling the discovery of new biological insights and accelerating drug discovery and development. Big data analytics. The integration and analysis of large biological datasets will provide deeper understanding of complex biological systems and drive personalized medicine and precision agriculture. Systems biology. Modeling and simulation of biological systems will predict cellular behavior and guide the design of synthetic organisms and bioprocesses. The future of biotechnology is marked by rapid advancements and interdisciplinary collaboration, driving innovations across diverse fields. From personalized medicine and regenerative therapies to sustainable agriculture and industrial processes, biotechnology will continue to transform our world. Emphasizing sustainability, health, and environmental protection, future trends will address global challenges and improve the quality of life, paving the way for a more sustainable and equitable future. 14. Ethical and regulatory considerations in biotechnology. Biotechnology holds immense potential for advancing health, agriculture, industry, and environmental management. However, it also raises significant ethical and regulatory considerations that must be addressed to ensure responsible development and application. Here are some key ethical and regulatory aspects in biotechnology. Ethical considerations. Genetic modification and editing. Genetic modification and editing, especially in humans, pose profound ethical questions. Techniques like CRISPR-Cas9 allow precise alterations in DNA, raising concerns about human germline editing. Editing human embryos can prevent genetic diseases, but also introduces the risk of unintended consequences and raises the prospect of designer babies, leading to ethical debates about eugenics and genetic inequality. Biodiversity. Genetic modification in crops and animals can impact biodiversity, potentially disrupting ecosystems and leading to unforeseen environmental consequences. Privacy and genetic information. As genetic testing becomes more widespread, protecting individual privacy and genetic information is crucial. Ethical concerns include informed consent, ensuring that individuals fully understand and consent to genetic testing and the potential uses of their genetic data. Data security. Safeguarding genetic information from unauthorized access and misuse. Discrimination. Preventing genetic information from being used to discriminate in areas like employment, insurance, and social services. Access and equity. Biotechnology can create disparities in access to advanced treatments and technologies. Ethical considerations include healthcare inequities, ensuring that biotechnological advancements such as gene therapies and personalized medicine are accessible to all, not just those who can afford them. Global disparities. Addressing the gap between developed and developing countries in accessing biotechnology innovations. Animal welfare. Biotechnology often involves the use of animals for research and production. Ethical concerns include animal testing, ensuring humane treatment of animals used in research, reducing suffering, and pursuing alternatives wherever possible. Transgenic animals. Using animals with altered genomes raises issues of welfare and the ethics of creating animals with specific traits for human benefit. Environmental impact. The release of genetically modified organisms, or GMOs, into the environment must be carefully managed to avoid adverse effects. Ethical considerations include ecological balance, ensuring that GMOs do not disrupt ecosystems, harm non-target species, or lead to loss of biodiversity. Sustainability. Developing biotechnological solutions that promote long-term environmental sustainability. Regulatory considerations. Safety and efficacy. Regulatory frameworks ensure that biotechnological products and processes are safe and effective for their intended use. Key aspects include clinical trials, rigorous testing through clinical trials to evaluate the safety and efficacy of new drugs, therapies, and medical devices. Risk assessment. Comprehensive risk assessments for GMOs, considering potential impacts on human health and the environment. Approval processes. Biotechnological products must undergo stringent regulatory approval processes before they can be marketed. Regulatory agencies involved include FDA, U.S. Food and Drug Administration, oversees the approval of drugs, biologics, and medical devices in the United States. EMA, or European Medicines Agency, regulates pharmaceuticals in the European Union. EPA, or Environmental Protection Agency, manages the environmental impacts of biotechnological products in the United States. USDA, United States Department of Agriculture, regulates GMOs in agriculture and ensures food safety. Intellectual property. Intellectual property rights, including patents, play a significant role in biotechnology. Regulatory considerations include patenting life forms, the ethical and legal implications of patenting genetically modified organisms, genes, and biotechnological processes. Access to innovation. Balancing the protection of intellectual property with the need to ensure access to essential biotechnological innovations, particularly in healthcare and agriculture. Labeling and transparency. Regulations often require labeling and transparency to inform consumers about biotechnological products. Key considerations include GMO labeling, clear labeling of genetically modified foods to allow consumers to make informed choices. Traceability, ensuring that biotechnological products can be traced through the supply chain to maintain transparency and accountability. International standards and cooperation. Biotechnology often involves global collaboration, requiring harmonization of standards and regulatory practices. Important aspects include international agreements, participation in international agreements and organizations, such as the Cartagena Protocol on Biosafety to manage the transboundary movement of GMOs. Harmonization of regulations, coordinating regulatory standards across countries to facilitate the global trade of biotechnological products and ensure consistent safety and quality. Ethical and regulatory considerations are integral to the responsible development and application of biotechnology. Addressing these considerations involves balancing innovation with safety, equity, and sustainability. Continuous dialogue among scientists, ethicists, policymakers, and the public is essential to navigate the complex landscape of biotechnology and harness its potential for the benefit of society while minimizing risks and ensuring ethical integrity. You have been listening to Biomedical Engineering, The Fundamentals of Biotechnology. Written by Phil Gilbert. Narrated by Kevin Brooker.